this is my first talk at the university, and the first time I'm talking about this project. So it was exciting presentation to make because it kind of showed me where I'm at and what I still need to do, which is slightly overwhelming, but it reframed everything for me, which was nice. But it also means I don't know how long I'm going to keep an eye on the clock because I don't know how much I'm going to ramble through these slides. So if I speed up or slow down, that's why. But yeah, I'll just go through my research background. So the Marie Curie postdoctoral fellowship is about finding a supervisor and coming up with a project that will hopefully get funded and goes for 2 years. But yeah, some of my research background will give some context around how me and my supervisor Rob Ogden came up with this project. So much of my research background has been in wildlife diagnostics. I got my undergrad and honours at University of Sydney, and that was kind of where I discovered this niche field called wildlife forensics. My honours project was about developing some genetic tools to identify rhino horn. So there's 5 species of rhino, and in a forensic case, you often need to know what species of rhino has been trafficked, and if it is indeed rhino horn. There's lots of fakes. There's horse hoof melted in the shape of rhino horn. There's buffalo horn that gets sold as rhino horn. So that was kind of where I got really interested in wildlife forensics, but diagnostics in general, what you can do with DNA, what questions you can answer. And so I sort of continued along that path, still at University of Sydney, focusing on these 2, probably my favorite species, these 2 cockatoos, the pink cockatoo and red-tailed black cockatoo. They're Australian species, and their evolution is quite complicated. For example, there's 5 subspecies of this red-tailed black cockatoo. So basically the project was trying to figure out what was going on, the phylogeography of these species, and then the applied side was these species get heavily trafficked, mainly into Asian markets. So they get poached in Australia as chicks or as eggs and trafficked around the world. So we wanted to know what populations were getting trafficked, and there was some illegal breeding as well going on in Australia. So there's a bit more fundamental biology in the PhD, but still with that applied diagnostic side. And then during my PhD, I started working for an NGO called TRACE, Wildlife Forensics Network. The director, who is my current supervisor, Rob Ogden, based in the vet building. And this NGO is basically promotes the use of wildlife forensics to law enforcement around the world, mainly focused in Southeast Asia and in Africa. And so I was based in Kuala Lumpur and Bangkok for a couple of years. Then COVID sent me back home to Sydney. And I'm still working for Trace on a part-time basis, obviously now in Edinburgh. And basically my role there was developing these diagnostic tools, again, focusing on wildlife forensics, and then also applying them. So yeah, I went to some crazy big sieges of elephant ivory, and these are probably 8 tons of pangolin scales. So I got to see what diagnostics at the front line looked like. And then I took a bit of a left turn for a while and did some postdoc work at the University of Sydney on cockroach genomics. And this is sort of where my friends lost interest in my work, because I was studying these really stunning species and super interesting. And then, but though it was a different study system, it was really fascinating to go back into sort of fundamental evolutionary biology. So we use these cockroaches and termites. So termites are just a type of cockroach, which I was mind blown when I learned that in probably 2020. But basically looking at the predictability of evolution, and there was a surprising amount of overlap between this work and my previous work on cockatoos and pangolins, because yeah, we're looking at the consequences of life history on genomic evolution. So termites are eusocial, cockroaches aren't, and looking at the genomic architecture that underpins key traits. So it's super interesting, but a bit of an aside on my main focus was being wildlife diagnostics, but I also got to learn a lot of bioinformatics during this postdoc. And then in October, which is what, 5 months ago now, I started a postdoc here. This Marie Curie project formed probably 2 years ago and got tinkered with, and yeah, I'm hosted by Rob Ogden over in the vet building. And I just wanted to linger on some of the molecular diagnostics work that I've been involved in, because it sort of gives context of some of the problems we're trying to solve in this postdoc. So first of all, yeah, I've talked about wildlife forensics. What it is basically the application of scientific analysis to wildlife crime prosecutions. So generating some diagnostic data around seized wildlife, and then basically using this in court to prosecute traffickers or poachers. I won't go in, so Rob's giving a talk on this. I'm plugging it on, what is it, Thursday night? Yeah, I don't want to talk about wildlife forensics the whole time, focusing on the actual Marie Curie project. But yeah, it's a field I really enjoy. And if you want to hear more about it, go and see Rob. But basically, mostly it involves species identifications in wildlife forensics. But more and more frequently these days, people need to know some more higher resolution questions. So identifying specific individuals, questions about parentage, geographic provenance. So if you have a sample, where did it come from? And this involves, I guess, a species ID involves usually 1 or 2 genetic markers, whereas these higher resolution questions, you need loads more genetic markers to answer them. And this requires a lot of R&D, basically a lot of species-specific assays that will be able to answer these more high resolution questions. So these are some of the projects I've been involved in that sort of questions beyond species ID. So cockatoos, we need to know some subspecies identification. One of the main projects I've been working on past 5 or so years is TigerBase, which is basically a DNA registry system of Southeast Asian captive tigers. Some work on pangolins, especially Sunda pangolins, which Nat here is also doing for his PhD, some more high resolution testing of them in Thailand. Some stuff on Asian elephants, black rhinos, lion individualization. I mean, the reason I'm showing these is just to show the great array of species specific tools we need to answer just a few of these high resolution questions. And it sort of brings in some gaps I'm trying to fill, or some challenges I'm trying to address in this Marie Curie project. Some disclaimers about this talk is, so I'm talking a lot about One Health diagnostics. Part of my fellowship is learning the pathogen diagnostic side of this work. So I've been surrounded by people that do pathogen diagnostics in the past, but haven't done it myself. So part of the project is learning that. So I might sound a bit naive when talking about some of the pathogen diagnostics work. There's lots of wet lab work during this project, and this is a computation genetics seminar, but I've been told that's okay. So, and yeah, it's 5 months in. I have some results, not a whole lot of exciting results yet. So this talk is going to be mainly about the problems that we're trying to address and the potential solutions, and a tiny bit of results. So in One Health, so I've been focusing throughout my research career in wildlife diagnostics, but this project is about moving this into how is wildlife diagnostics relevant to human and domestic animal health? And so, and the second point is, wildlife diagnostics is often about the host, the wildlife itself, or the pathogen. It's not usually both. In some studies, you see them both combined, but usually these two fields sort of act in isolation. But they are usually manifestly relevant to one another. So if you know a bit about the pathogens specific wildlife individual harbors, it's very good to know about that specific wildlife as well, and vice versa. I'll give some examples later. So wildlife interactions with domestic species and humans are increasing, and the world's becoming very globalized. Human population growth is huge, and climate change is changing things, and there's increasing international wildlife trade. So animals are basically moving everywhere. There's lots of humans everywhere moving stuff, and it's all becoming sort of a little bit out of control. So there's just loads of interactions between wildlife and domestic species and humans. So this increases the potential for pathogen spillover events. I mean, I think we all learned a lot about this sort of thing since COVID. I certainly did. Yeah, how prolific zoonosis could become in today's context. So back in 2008, it was estimated that 70% of emerging infectious diseases in recent decades originated in wildlife. I'm not sure what that number looks like now, but basically, yeah, a lot of diseases we're having to deal with in the world today emerge from wildlife. And this increased globalization not only increases spillover events, but increases the severity and speed of these outbreaks. So I'm from Australia, where we have such tight biosecurity. If you've been there, the biosecurity at the border is, it can be a real drag getting anything in. And yet we're still not able to keep these global diseases out. There's just so much movement around the world, via air, via ships. So yeah, part of the project is looking how wildlife diagnostics can inform One Health issues. So we've got human diagnostics here and also domestic animal diagnostics, and sort of this overlap between the three, and between wildlife diagnostics and the other two, is what I think is fairly untapped at the moment. Again, I think a lot of these fields operate in isolation, and this seems to be changing. There's a lot of movement around One Health, but it's still an issue. So how can wildlife diagnostics inform human diagnostics and domestic species diagnostics? I think it's pretty intuitive. I mean, surveillance of emerging diseases and identifying threats to domestic species and humans, that's just two examples. But yeah, I think, are we going through some more examples? I think next slide, if I can change it. Yeah. So these are just some examples I've seen personally, and there are loads more out there, but I think it sort of just shows how manifestly relevant wildlife diagnostics can be to these other arenas. So when the Sunda pangolin was first implicated in COVID, it was thought to be where COVID originated, which was later disproved. But yeah, the main paper that showed these Sunda pangolins had coronaviruses never said anything about where these pangolins were from. There was nothing about the pangolins themselves. So they harbored all these coronaviruses, which may have caused COVID. And the reason they didn't say anything was possibly because it was a disease lab and not a wildlife forensics lab or a wildlife diagnostics lab, or maybe because they didn't have the capacity to do this testing. We at the time at Trace had unpublished reference data, and we were able to find out where these specific pangolins came from, and it looked like they came from the North Borneo region. Now, whether these pangolins actually contracted these SARS-CoV-2 viruses in North Borneo, or whether they were trafficked to China and then got the virus, I mean, we'll never know. But I think it's a good example of where host diagnostics could have really helped uncover a worldwide I mean, they didn't end up being the source of SARS-CoV-2, but yeah, they still carry lots of viruses, and they're seized every day, and yeah, they have the potential to spread these diseases to other pangolin populations or other domestic animals or even humans. Another case in Australia there was some prawns seized with white spot disease. Usually these would go to the wildlife forensics lab to find out what these prawns are, where they came from. But because they had the disease, the customs officers saw that they had white spot disease. They got sent to the disease lab instead. And so they did some disease diagnostics on these prawns, and they never made it to the wildlife forensics lab to find out what these prawns were, where they came from. Again, combining these data would be very, very valuable in this situation, but it just doesn't seem to happen. There's just too much divergence between the fields in most settings. This is an example closer to home. I know the vet school has been working a lot on scimitar, horned oryx. So they get translocated as part of their conservation management. Knowing about their genetic diversity plus their pathogens is kind of crucial to these translocations, right? So they harbor these hemoparasites, and the movement of these hemoparasites plus the genetic diversity of the scimitar horned oryx has to be taken into account at the same time in a sort of combined framework for these conservation translocations to be successful. And lastly, again, one closer to home, I think a lot of people at Roslyn are working on avian influenza spread by, the wild species are sort of, I don't know if were they the originators of the current outbreak. They certainly helped the disease spread around the world, and sort of monitoring wildlife plus the avian influenza variants in a combined framework, again, seems to be the right approach. And again, it doesn't seem to happen too much. Now, I will say in some labs, in some institutions, it can happen. There are enough scientists around that they talk to each other enough. Sometimes it gets done in the same lab, but in many contexts, it doesn't happen. So some of the key challenges we face of why this might not happen is non-standardization of diagnostic approaches. So much specific equipment and specific reagents and assays are needed per species and per pathogen. And I'll give some examples. So these are some of the projects I worked on that I spoke about previously. And these are the different markers we use and the different equipment we use to generate these profiles from these species. So a lot of the labs we work in Southeast Asia and Africa, they're not going to have all this equipment in one institution. I mean, we're lucky at the Roslyn Institute. It's huge. There's lots of people working here. We might have this. So there's a 12K flex machine, open array chip, real-time PCR machine, bioanalyzer, MySeq. So my point here is that every species or every project tends to use, well, it certainly uses a different set of reagents or assays to generate the relevant data, and then also often different equipment. And there are many others beyond this. I haven't even gone into the pathogen diagnostic platforms and assays. And then having so much equipment and so many different assays and workflows, it complicates the validation of these workflows, and the amount of training required was to be able to do all of these examples: tiger base, Asian elephant, pangolins, black rhino. The amount of infrastructure they would need, the amount of training they would need is very challenging. So therefore, diagnostic labs often have limited access to numerous species, pathogen-specific tests. So this is kind of what I mean by the difficulty of everything being quite non-standardized. And this is just basically because technologies evolve. Another key challenge related to this is the considerable R&D time for each system. So every time you have a new set of questions or a new species you need to work on, basically it can take years to develop. It takes a whole PhD, or tiger base, for example, from its conception took 10 years from the very start. about 3 years of validation experiments and building capacity. And it's just not doable for the thousands of species and pathogens we need to consider, especially the combinations of species and pathogens. So you might have a test for lions and one of its key pathogens, but then you need a test for its other key pathogen. It's basically just unfeasible, especially because diagnostic requires considerable quality control considerations. So especially forensic validation takes a long time. So the cost and time of R&D limits accessibility to what some people call day zero diagnostics. So the day a pathogen is first discovered and an outbreak is first discovered, the R&D it takes to then design a diagnostic test to test for this, it's often too late by the time it's up and running. These emerging threats are super time sensitive. Emerging issues, so similar to the above, especially the wildlife trade dynamics changes so rapidly. So we developed tiger base because of trade in tiger parts. There's a lot of trade in tiger bones, but tiger bones were becoming rarer and rarer, and these sort of started getting replaced by lion bones. So now when we see big seizures of what they say are tiger bones, they're actually lion bones. So now we're working to create a system for lions, which takes a whole other few years of R&D. And then developing panels for relatively rare cases. So if it takes a few years of R&D, is it worth your time and effort if this question is not going to pop up every few days, every few weeks? So we see this a lot with bushmeat cases, especially in Africa. There'll be a case, a good example is there'll be some hippo meat or a hippo carcass and a machete with some suspected hippo blood on it. And we want to be able to use a profiling system to match that hippo carcass with the machete blood to sort of reconstruct a crime scene. But this doesn't happen very often. And for us to do this, we'd have to create a hippo panel, pick whatever platform we want to put this on, validate it. It's a lot of work. And the bushmeat species might not be hippo, it might be warthog. So yeah, it basically might not be worth the cost and time it takes to develop, considering how long the R&D takes. And this is, one of the key challenges I mentioned a couple of slides ago, that pathogen wildlife / host diagnostics typically operate in isolation. And then these diagnostics certainly operate in isolation from domestic and human diagnostics most of the time. And I used the example in Australia with the prawns. Often agencies that need these diagnostics, like customs or people in the field, will choose one or the other. On occasion, they might be lucky and the facility they send it to has capacity to do both. Or if they're really good, they'll send a sample to each and get this combined information. But it doesn't happen too often, unfortunately. And so therefore there's often no co-analysis between the wildlife and pathogen diagnostics, even though they're so relevant to one another. So how do we combine them in a One Health framework to encourage this interdisciplinarity and co-analysis? So the overall goal is to develop and optimize adaptable molecular wildlife diagnostic tools. Adaptable meaning one tool can address lots of questions or lots of species. So we're not constantly having to develop new projects, perform new R&D and new validation experiments every time we have a new question. And then so they're also standardizable and adaptable across different diagnostic sectors, whether it's pathogen diagnostics or wildlife host diagnostics. And these objectives, they seem to change every month. They're certainly different from the project conception. They're similar, but they've become sort of broader. But basically leveraging Oxford nanopore technology sequencing advancements, which is used a lot in the Roslin and at the vet school, to develop these adaptable workflows, and then develop some streamlined bioinformatic and statistical tools to simplify and standardize the wildlife diagnostics, and then sort of a broader challenge, more of a desk-based project, is evaluate approaches to combined pathogen and host diagnostics. And I haven't mentioned red squirrels yet. So this is the study system I'm using for all of this work. And I'm hoping that the work can be beneficial to the red squirrels and squirrel pox situation itself. And I'll explain a bit about the red squirrels soon. But first of all, so we're quite excited about Oxford nanopore technology sequencing, especially their recent advancements. And yeah, I was talking about a standardized platform earlier. We're excited that this could be it, or at least emerging upon something that can be used to answer a diversity of questions. So that some of the key requirements we need for an adaptable platform is obviously it needs to be affordable and accurate to be realistically used on the front line. The resultant data needs to be transferable across labs. So a lot of the older sort of diagnostic platforms, data you would create in the run lab wouldn't be able to be used in another lab without some calibration and a lot of standardized samples across the lab. And the technology needs to be flexible in the type and number of genetic markers. So yeah, well, I'm certainly excited about the MinION advancements over the last 5 or so years. It's become a lot more accurate and simple to run. So the MinION is basically a portable Oxford nanopore sequencer. It produces data in real time. And yeah, the way it works is has a membrane with lots of channels on it, and the DNA molecule passes through these channels. It disrupts an electric signal and creates this sort of squiggle, and the squiggle is then decoded into a DNA sequence. As I said, it's in real time. So it's relatively inexpensive, which is great for diagnostics. It's sequence length agnostic, so it can sequence short, long markers, whole genomes, amplicons, which makes it quite flexible in the number and type of markers it can sequence. And as I said, the recent advancements, so I used a MinION close to 10 years ago now, and the library prep took all day. The error rate was fairly poor, but yeah, as of the last few years, this has completely changed. It's almost as accurate as Illumina, and the library preps are a lot more simple and more robust. And importantly, it's been the use of MinION and Oxford nanopore sequencing in general. It's proven for use in wildlife forensics and in pathogen diagnostics here as well at the Roslin and Vet Institute. So I think a lot of people at the Roslin are certainly excited about it. They're using it a lot for disease diagnostics. I know Rob and his research group are using it a lot for wildlife forensics. So I think it's potentially a really good platform to start standardizing a lot of these assays. So one of the cool functionalities of ONT sequencing called adaptive sampling is something that I want to try as part of this Marie Curie project. So I'll try and slowly, it's much easier with a video explaining what adaptive sampling is, but basically when your DNA molecule is being sequenced through one of these channels, it sequences for about a second and creates a chunk of sequence, which is about 400 base pairs. Then this adaptive sampling software basically makes a decision on whether this sequence is a sequence you want or don't want, And if it's something you don't want, the membrane will initiate a voltage reversal and spit out that molecule and try the next one. I think my next slide explains it a bit better than this one. Creates this chunk, 400 base pairs of sequence, and basically you give the software some reference sequences you want the sequencer to target. So you might give it, say, 50,000 base pairs of reference sequence within your 3 gigabase pair genome, and you say anything, if you're sequencing anything from this reference database, please keep sequencing the molecule. If not, stop, reverse the voltage and spit it out and try the next molecule. So it basically allows the sequencer to enrich your regions of interest and deplete regions you're not interested in. And we're terming that, I don't know if it's been used before or whether it's just something me and Rob use, digital enrichment. So it basically requires no biochemical enrichment. You're basically giving it the raw DNA, and the software is doing the enrichment during the real-time sequencing. As well as targeting specific sites, you can deplete specific sites. So this is super useful if you want to do some host depletion say you've got a DNA sample with a pathogen you're interested, it's going to have a lot of host DNA in it. You can tell the adaptive sampling software, I want to get rid of the host DNA, and it will actively chuck away those DNA molecules. And you can also use it for barcode balancing. So if you have, say you're running 10 different samples in the same run, you can tell the software that once my sample gets to a specific depth, stop sequencing that sample and concentrate on the other 9. And then it sort of balances out how much coverage you're getting between your samples. So using this, I mean, coming back to the theme of adaptable tools, if this worked perfectly, labs may not even need species-specific assays. So they could use the same sequencer, the same library prep for every scenario they work with, for every species, every question, and just alter the adaptive sampling targets. That would be the dream. They just need one platform to do everything, and it's completely flexible. Any number of type of targets you can specify. Yeah, but unfortunately it doesn't work as well as we would like it to. So, I mean, I think anyone that first reads about this gets super excited about how cool it could be. I certainly did. But yeah, how well does it actually work across different diagnostic scenarios? And yeah, word around town is it doesn't work as well as people have had mixed feelings about this technology. So part of my Marie Curie is, where can it help, and can it be the savior of standardizing wildlife diagnostics? Now, I don't think so, but it can certainly help in some situations. So a bit about the study system. So the red squirrel and squirrel pox is sort of an active One Health diagnostic situation. So the red squirrel is endangered in the UK, and that's partly because of, well, a key factor is the spread of squirrel pox. And the grey squirrels are thought to act as a reservoir for this spread of squirrel pox. And the vet school are doing a lot of work. There's a big red squirrel group, and red squirrel carcass is seemingly everywhere. Ongoing mortality surveillance around the vet school to figure out what's going on, where this is spreading, how the red squirrels are faring, different populations. And so yeah, it's an evolving situation. Squirrel pox first emerged in the south of Scotland in 2007, and the red squirrel group here at the vet school in Roslyn found that it had spread north, I guess last year. But this paper was published in 2024. I think a case was found at the start of last year it is worryingly spreading north of the central belt. So it's an evolving situation, and really worrying. And Melissa's paper came out a few weeks ago, which not only to red squirrels have this threat of squirrelpox, they have a super low genome-wide heterozygosity. So the ability to respond to this new virus and to different fragmenting habitats is likely not going to be great. The translocations and sort of ongoing conservation management of these squirrels is going to be a lot of work and take a lot of diagnostic information. So this is where I hope my project can contribute to. There's a lot of ongoing squirrel work going on, and I'm hoping to be a small benefit in a small way by sort of using it as a study system, but also contributing to the diagnostics of red squirrel and squirrelpox. So how we can help with the red squirrel itself, host diagnostics, is to ongoing monitoring of genetic diversity, especially if there are going to be translocations, but also looking at some specific genes within the red squirrel genome, such as MHC and other immune-related genes, and how they might underpin different responses to squirrelpox. And then for the squirrelpox itself, monitoring the spread and trying to figure out the evolution of this virus. So looking at the different variants of this virus. So at the moment, there's been a lot of presence-absence testing. So does a squirrel have squirrelpox or not? But what isn't known is, are there different variants contributing to this outbreak more so than other variants? What are the characteristics of this virus that are contributing to their virulence? Just going to grab some water. And my theme of combining host and pathogen diagnostics, how does the combination of these two help with this very complicated red squirrel situation, which I'm constantly learning about, trying to decipher this map with the different regions that the red squirrels are found in. There's a lot of genetic structure in the red squirrels, and yeah, as I said, the ongoing situation of this squirrelpox moving north becomes quite complicated from a diagnostic perspective, and being able to combine the red squirrel diagnostics with squirrelpox, I think will benefit our knowledge of what's going on at least a little bit. And it will hopefully be a good model system for assessing these adaptable diagnostic approaches. So I'm going to speed up a bit now. So I'm basically going to be testing with the squirrel and using simulations 3 adaptable diagnostic workflows based on MinION sequencing. So the first one is super adaptable because all you need to do is give the MinION some raw DNA and do some whole genome sequencing, and then extract your markers of interest from there. It's obviously very expensive, and you're producing a lot of redundant data. The second is multiplex amplicon sequencing. So using specific biochemistry primers to target your genes of interest, amplifying that, then sequencing them. This obviously relies on you to have a species-specific markers, which is not so adaptable, something I'm sort of looking to avoid to make these more adaptable approaches. And then there's adaptive sampling, which I've just gone through. So this doesn't need any biochemical enrichment, and hopefully you're not producing a lot of redundant data you don't want. So in the first scenario, we're producing a lot of DNA sequence data and doing a lot of bioinformatics to pull our markers out. Second scenario, we need this biochemical enrichment step, which is going to be, in this case, specific to red squirrels. It's not going to work on any other species. And this third scenario would be ideal, but we don't know how well adaptive sampling actually works in a lot of cases. Yeah, so just basically been through that. This whole genome sequencing requires loads of sequencing that's going to be predominantly redundant, especially if your pathogen occurs at a really small portion of the DNA in your genome. So squirrelpox in a DNA sample is going to be less than 0.01% of the DNA probably. So you need a lot of sequencing to get the squirrelpox genome out. And how low we can go is I think a good question. So how little sequencing can you produce to get your answer? Some work by one of Rob's students last few years has tried this with geographic provenance applications, and she found that in some scenarios you just need 0.5 times coverage to get the information you need. I was looking at this recently from the perspective of trying to match individual samples together. So if you have really low coverage, so you have 2 times coverage, most of your sequence coverage is going to be around that 2 times mark. But there will be, by the stochastic nature of sequencing, some higher coverage across the genome. Can we use that to overlap it with our matching samples to generate some reliable SNP matches? So again, our hippo scenario, can we generate some whole genome sequencing information from the hippo and from the blood on the machete, find those high coverage overlapping SNPs and get a match? I did this last week with just some 2 times coverage sequencing from 2 separate libraries from the same hippo, and found 16 really high quality overlapping markers suitable for match probabilities. So this is possible with pretty low coverage. But the more samples you compare, if we're trying to match up, say there was other samples like some hippo meat, some hippo tusks, the same crime scene. If we wanted to create libraries from all these different samples and combine them and co-analyze them, we're going to see less and less overlap between the SNPs. So this is sort of, this is ongoing work, and it brings me a point I'll talk about later about a new statistical framework that we've developed to match these different samples. So the second one is multiplex amplicon sequencing. This is actually what we're using mostly now on the MinION. So it's very simple. Basically, you have your marker. We're typically focused on SNPs, and you have your forward and reverse primer a little way around the marker. So we're typically using fragment lengths of about 200 base pairs. And then we can basically multiplex many hundreds of these SNPs in the same PCR and then sequence them on the MinION. It's amazing the multiplexing capacity these master mixers these days have. So for a Black Rhino project we're working on now, we can get 196 SNPs in the same single master mix and multiplex them, and it seems to work quite well. So yeah, this approach obviously requires species-specific biochemical enrichment, but once it's developed, it's very cheap to run this. So all you need is your primer pool, and it'll enrich obviously the exact regions you want, and then sequence it, and you've got your answer. So yeah, as I said, this is currently what we're mostly using now. So we've got projects at the moment, hopefully a manuscript in the next few months, developing this multiplex amplicon sequencing assay for tigers, cheetahs, and tamarins, and then a Black Rhino project that's sort of in the works still, but it's creating some great results with this platform. On the pathogen side, there's similar platforms for doing multiplex amplicon sequencing to generate the pathogen genome using tiled sequencing. So again, you're multiplexing lots of amplicons in the PCR, tiling these amplicons around the genome to produce the entire genome sequence. And they've done this a lot with SARS-CoV-2, and then here at the Roslin also for African swine fever virus. So this is obviously a great approach, one that's being used a lot now, but it still does require that species-specific biochemical enrichment step. And yeah, basically, we want to try this on the red squirrels to get that diagnostic information we need. So whether it's the MHC region, fragmented parts of the genome to get some genome-wide diversity, and testing some other sort of more adaptable ways of producing this information. So there's some cool approaches such as ISSR-seq, where maybe you don't need species-specific primers to generate these amplicons. This approach uses basically these short sequence repeat motifs as primers and targets random parts of the genome for each species, and then sequences the part in between. So in this image down in the bottom right, you've got this example of a GAA times 6 motif. So the GAA times 6 is going to be found throughout the genome of every animal genome, basically. And then you do some competitive PCR, generate some amplicons, and sequence them. So this way you could possibly use these sort of really redundant primers for lots of different species. So it does become a more adaptive approach than your species-specific primers, lowering that R&D time, basically. And then, of course, what I think is going to be the most realistic scenario is using this multiplex amplicon sequencing in combination with adaptive sampling. So for example, doing the ISSR-seq method with some barcoding balancing via adaptive sampling, or some host depletion via adaptive sampling. I think that's where we're going to basically land on at the end of this project. So adaptive sampling, what experiments I want to run with the red squirrels, is basically consider all the things that might impact your efficacy of adaptive sampling. So it's unlikely to work well if your targets are less than 0.1% or more than 10% of the genome, because the pores get quite tired of the reverse voltage and spitting out the molecules, and the pores get blocked, and basically your overall yield goes down. So things like how many nuclease flushes should we incorporate during the library, during the sequencing. You can see this nuclease flush improves the yield dramatically halfway through the run. Looking at shearing the DNA, which actually helps in the context of adaptive sampling, because it means your targets can be shorter. The use of buffer sequence to ensure that your chunk that gets aligned to your reference sequence as part of the adaptive sampling process. If it doesn't align to the target sequence but aligns just outside of it, if you're adding some buffer sequences, it will make sure that your target sequence is still sequenced. And there's loads of adaptive sampling algorithms out there now. So yeah, it seems every year there's 2 or 3 papers about some new and improved adaptive sequencing algorithms. So it's sort of an evolving field. So which ones work the best is something I'd like to test using the squirrels. And then basically we want to just see how much enrichment will this adaptive sampling give us, and then therefore how much sequencing is required to give us these reliable diagnostic results. And then comparing it to the other 2 workflows I just went through. So yeah, for the red squirrels, the experiments I'm hoping to run in the next few weeks is targeting some genome-wide markers, whether it's microhaplotypes or just random non-coding parts of the genome, and this is for genetic monitoring. Some more targeted markers for MHC and other immune-related genes to correlate how these different squirrel populations or individuals respond to the squirrelpox virus. And then for the pathogen itself, for the squirrelpox, trying out host depletion. So basically, we've got a DNA sample which is mostly squirrel. What happens if we sequence it and tell adaptive sampling to not sequence the squirrel DNA? Will that work for the squirrelpox? And then, yeah, the other way around is specifically targeting the squirrelpox genome, which will mean that chunk alignment will be a lot faster. And this really depends on how much squirrel versus squirrelpox DNA is in our samples. And that's kind of a lot of the work I've done so far to figure out that exact question. So I've created some squirrelpox cybergrain tests to figure out the copy number of squirrelpox in the sample, and doing some host depletion during the DNA extractions to see if I can sort of during the DNA extraction get rid of some of that host DNA, the squirrel DNA, and slightly enrich the squirrelpox DNA, and then designing a quantification workflow to sort of decide, is it worth sequencing? So if we have a squirrel sample and we know that there's less than 0.0001% of squirrelpox DNA in there, it's probably not worth trying adaptive sampling or whole genome sequencing. But we basically need to figure out, most of my work the past few months is creating this quantification workflow by setting up some standard curves and using single copy genes to figure out copy number of squirrel genomes in the sample versus copy number of squirrelpox genomes in the sample, and then correlating that with your read output. So if we find that squirrelpox DNA in the sample comprises about 1% of the DNA, actually lead to 1% of the read output when running it on a MinION. And again, to keep with this adaptable approach, I'm using really conserved single copy mammal genes for this quantification, so that hopefully that is it worth sequencing quantification approach can be used for other species as well. And yeah, I was lucky that sort of when I designed all these experiments, it happened before a couple of really useful pieces of software came out. So I can simulate a lot of this adaptive sampling and a lot of these experiments. So again, this is what I've sort of been working on the last few months, to sort of simulate as much as I can, find sort of the best scenarios as possible, and then that's what I'll be running in the lab. Okay, this is only 2 slides, this objective 2.
A few more minutes, so we have some time to ask you questions.
No worries. I'll skip through this slide on to the next slide. I'll mention wildlife detector because it's something Emily and I have been working on. This objective 2 is about basically simplifying, when you've got your MinION data, how do we turn that into a diagnostic result that can be interpretable, and how can we standardize this? So it's using basically bioinformatic pipelines and software to sort of wrangle this into something that's actually useful. And a few weeks ago, Rob and I stumbled upon a potential approach to revolutionize is a strong word, but we are very excited about it, to sort of tinker with how we do match probabilities. So match probability is when you're trying to do an individual identification of something in a diagnostic scenario. You need to know how likely it is to observe this same profile in the general population. So I'll give an example with the hippos. If we've got 3 loci we're matching between the hippo and the blood on the knife, I mean, how can we be sure that this is the same individual? We really can't be with 3 loci. There's going to be loads of hippos in the population with the exact same genotype across these loci. But if we have 1,000 matching loci, obviously there's a very, very high chance that these are indeed the same individual and not just some other random individual in the population. It's really crucial for forensics, but also for other applications, such as invasive species management and individual genetic monitoring. Genotype match probabilities rely on huge population genetic data sets because you need to estimate realistic allele frequencies. But what we're excited about, again, this is something we're working on right now as of only a few weeks ago, is we're developing a novel statistic approach for calculating match probabilities in this exact hippo scenario without the need of any population genetic data. And therefore any marker system could be used to do this. So it's sort of the ultimate adaptive approach to match 2 samples in an individualization scenario. But we're still checking the numbers, so I won't actually present our workflow there. This is work in progress. I never thought I'd get to this, so I'm going to skip right through it. And thank you. So I didn't know about the timing. As I said, this is the first time I've presented this work. So It was almost on time. You didn't miss out much. The last objective I've hardly started. But yeah, thank you for listening, and thank you for having me along to the talk.
Okay, thank you. Do we have any time for questions? Any questions in the room?
So initially, like at the start of your presentation, when you talked about the different projects and the different systems and SNPs you're using and so on, there were for some species was mitochondrial DNA used and part of genomic DNA. Is there a reason?
Yeah, so I mean for some, I'll repeat the question for those online. For some systems we're working on, we're using mitochondrial DNA to generate this diagnostic information rather than nuclear genome-wide SNPs, for example. The main reason we're doing this is because it's cheap and easy. It's going to give more crude information. So, for example, the Sunda pangolins, at the moment, we're using mitochondrial DNA to basically tell us if they're from North Borneo or not from North Borneo. And Nat's PhD work is analyzing lots of Thailand Sunda pangolins and using genome-wide SNPs, and you can get much higher resolution than this. But then to roll out those SNPs in the field, you need to make sure the labs using this test have the right equipment and the right assays to actually carry it out. So we tend to start, I mean, a lot of the time, mitochondrial DNA gives you enough information to answer the question, but you can always get more out of genome-wide SNPs, but more expensive and more R&D.
Okay. So I was wondering, for your objectives, you were thinking to use the biochemical enrichment and adaptive sampling. How many markers do you expect to need to use in these cases? Like, are you just trying to identify, okay, it's a squirrel, or it's not a squirrel, or it's a fox, or it's not a fox?
Yeah, so I think that's one of the key questions, actually. So we don't need that many markers to answer our question. Yeah, if you want to be able to track individual squirrels, you could probably do that with, say, 500 markers. However, that might be apparently that's too few for adaptive sampling to be effective, because you need to target at least 0.1% of the genome. Otherwise, the pores get tired and they get too blocked because you're rejecting too many molecules. But I've also spoken to people that said it probably would work. So I think part of the experiments is, it can be completely flexible how many we use. I mean, if we use 500,000 markers, it'll be nice diagnostic information, but will it be cheaper and easier to target 500 markers?
So this leads to my next question. Do you have a price tag on these, like for 500 markers or 500,000 markers?
Not
to give us a ballpark.
Yeah. I think that's completely dependent on how much enrichment we're getting. So if it's perfect enrichment, it will be as expensive as a multiplex amplicon PCR test, but it won't be. It's what we might. This is part of what I'm trying to test. How much enrichment can we get, and therefore how much can we lower the price to actually make this applicable in the field? I have a feeling the enrichment won't be good enough, speaking to people, and it might be more of a day zero tactic. So use this until you can create a PCR amplicon assay, which will then be much cheaper and easier to run. But that's exactly what I'm trying to find out.
Okay, thanks. Do we have any other questions online as well?
There's nothing in the chat.
Okay.
Yes, please. So I think the latest approach you were talking about, it's not based on population statistics. Can you at least tell us what it's based or what's the idea behind it?
Yeah, I think I mean, it's not secret. It's just we didn't have like, we want to check everything first. But basically, it's more of a paradigm shift than anything, because when they created, over the last 40 years, when they have been tinkering and developing these match probability statistics, it's been based on relatively small set of markers, so usually STRs, or if it's SNPs, not that many SNPs. But now we have lots of sort of even medium throughput panels, so 100 or a few hundred SNPs or more. We can be completely conservative about how we assume that allele frequencies to be. And so we may not need to actually estimate them, which is the point of the population genetic data. We can take the worst case scenario and have this medium throughput panel, and it still has plenty of power to answer our question. But yeah, I'm pretty confident it's it's all going to work out. But I was speaking to Rob this morning, and we decided it's probably too early to present any more details, just in case we're completely wrong about this and it falls on its face.
I just wondered if you had any plans to also use grey squirrels in your work. I think we'd assume that it's the same strains of squirrel pox viruses circulating in both reds and greys. But I don't think anyone's checked.
Yeah. No, I think that would be, I mean, getting any information about the variants that are floating around in greys and reds would be super interesting. How to analyze that, that's beyond me at the moment. Something I'm hoping to learn, but yeah, basically a lot of this is inspired about how they did the contact tracing during COVID. So much information was brought together about who had what strain, where were they at different times, and combining all of this information and how it spread from this population to this population. I think a similar thing could be done for the squirrels. Do these grey squirrels have this variant, and how did it make its way to this red squirrel population, and then how did that spread to this red squirrel population? It might be super complex scenario, but we can slowly uncover exactly what's going on the more diagnostic data we chuck at it. So yeah, I would be interested if it all works well and there's extra money to do grey squirrels as well.
All right. Thank you very much, Kyle, and thank you everyone for joining us today.
Thank you for having me.